A+ലേക്കുള്ള വഴി ഇനി തനിയെ തുറക്കാം. Subscribe to the ENCLAPS channel. Ring the bell button and open success. It's that easy.
Welcome to the fascinating world of biology. Hybridization experiments conducted by Gregor Johann Mendel in the pea plant laid the foundation of genetics. He observed that all plants obtained by hybridizing tall and dwarf plants resulted in the production of only tall plants in the F1 generation. The fact that no intermediate plants were formed by the hybridization of tall and dwarf plants and the factor responsible for dwarf remaining hidden persuaded Mendel to continue with his experiment. He self-pollinated the first generation plant to understand what happened to the factor responsible for dwarf in the first generation. Now we can complete the illustration. Plants in the F1 generation have both the alleles responsible for the characters tall and dwarf. During gamete formation, these alleles separate and move to different gametes. Similarly, gametes formed from the second plant also produce gametes with capital T and small t alleles. What happens when these gametes get fused? Capital T and capital T combines to form capital T, which is responsible for the character tall. Capital T and small t combines to form capital T small t, which is responsible for the character tall. Plants raised by self-pollinating the F1 plants are called F2 generation plants. Can you find that the first three plants given in the illustration are tall and the final one is dwarf? From this experiment, it is clear that the character dwarf that remain hidden in the first generation become visible in the second generation. Of the 1064 plants obtained in the second generation, 787 plants were tall and 277 dwarf. The ratio of the result obtained is approximately 3:1. Mendel continued his experiments based on contrasting traits of other 6 different characters in the pea plant. We can complete the table and compare the ratios. Parent plants, first generation, second generation. Flower position may be axial or terminal. In the second generation, 651 are axial and 207 are terminal. So, the dominant character is the axial flower. Seed shape may be round or wrinkled. In the F2 generation, 882 are round and 299 are wrinkled. So, the seed shape of F1 generation plants are round. The ratio obtained in both cases is 3:1. How recessive character in the first generation gets expressed in the second generation? Mendel assumed that during gamete formation, the factors that determine a particular character segregate without getting mixed. That is, half of the gametes formed from first generation plant capital T small t contain capital T and the other half contain small t. The inferences formulated by Mendel from such experiments are given below. A character is controlled by the combination of two factors. One trait is expressed, that is the dominant trait, and the other trait remains hidden, that is the recessive trait in the offspring of the first generation. The traits that remain hidden in the first generation appear in the second generation. The ratio of the dominant and the recessive traits in the second generation is 3:1. In his first experiment, Mendel observed the inheritance of a single pair of contrasting traits. He observed the inheritance of 2 pairs of contrasting traits of the same plant in the next stage. Observe the illustration showing the hybridization experiment conducted by Mendel on 2 characters, namely height of the plant and shape of the seed. Parent plants, tall round seeds, capital T, capital R. Dwarf wrinkled seeds, small t, small r. The gametes, capital T, R, small r. First generation, capital T, capital R, small r. While self-pollinating the first generation plants, capital T, capital R, small r and capital T, capital R, small r, 4 types of gametes are formed from each plant. Which are they? Capital T, capital R, capital T, small r, small t, capital R, small t, small r. Now, we can make a table for listing the gametes formed from these 2 plants and the F2 progenies formed during the fusion of these gametes. capital T, capital T, capital R, capital R. Tall, round seats. Capital T, capital T, capital R, small r. Tall, round seats. Capital T, small t, capital R, capital R. Tall, round seats. Capital T, small t, capital R, small r. Tall, round seats. Capital T, capital T, capital R, small r. Tall, round seats. Capital T, small t, small r, small r. Tall, wrinkled seats. Capital T, small t, capital R, small r. Tall, round seats. Capital T, small t, small r, small r. Tall, wrinkled seats. Capital T, small t, capital R, capital R. Tall, round seats. Capital T, small t, capital R, small r. Tall, round seats. Small t, capital R, capital R. Dwarf, round seats. Small t, capital R, small r. Dwarf, round seats. Capital T, small t, capital R, small r. Tall, round seats. Capital T, small r, small r. Tall, wrinkled seats. Small t, capital R, small r. Dwarf, round seats. Small t, small r, small r. Dwarf, wrinkled seats. Through this experiment, Mendel could find the reason for the appearance of new combination of characters in the offsprings that were not expressed in the parents. Mendel presented the most scientific inferences about the inheritance of characters. However, at that time, the scientific world discarded his inferences. Scientists named Carl Correns, Eric von Schermar, and Hugo de Vries conducted similar experiments and observations independently. Before publishing the results of their studies, they noticed the experiments and inferences of Gregor Mendel. Then, they published their study results to substantiate the inferences of Gregor Mendel scientifically. Thus, in 1990, the inferences of Mendel were established as Mendel's law of inheritance. Mendel explained that the appearance of variations in offsprings, that is, the characters not present in the previous generation, is due to the independent assortment of each character. Experiments conducted by Mendel and the inferences obtained from it form the building stone of genetics. Let us meet in another exciting class of biology.
3 ലക്ഷത്തിലേറെ വരുന്ന ചോദ്യോത്തരങ്ങൾ ഏറ്റവും കുറഞ്ഞ ചിലവിൽ സ്വന്തമാക്കുവാൻ ഉടൻ തന്നെ ഡൗൺലോഡ് ചെയ്യൂ എൻക്ലാപ്സ് ദ ലേണിംഗ് ആപ്പ്.